example. It is included in your syllabus, so we need to discuss it. So next is prokaryotic cell. So the cell which does not have a the type of cell which does not have a nuclear membrane and other membrane bound cell organelles, this is called prokaryotic cell. Example কি prokaryotic cell এর example তোমরা জানো তো?
Bacteria.
Bacteria, very good. So prokaryotic cells are placed in the kingdom Monera. আমরা already পড়েছি. So the cells represented by bacteria, then different kinds of cyanobacteria, that means blue green algae, and most common type of organisms. These are most common type of prokaryotic organisms. So these prokaryotic organisms, that means cells, are generally smaller and multiply more rapidly than the eukaryotic cells. There are some differences between prokaryotic and eukaryotic cell. First, we need to know the detailed structure of prokaryotic cell. So let's see the structure of a bacterial cell, then we will understand the differences. Acha, can you see the picture? Can you see the picture, Trishita?
Yes, ma'am.
Okay. So it is a bacterial cell. Hmm. So bacteria is a prokaryotic cell, and all prokaryotic cells are similar in their organization. Though their shapes uh, and their functions are different, but there is a similarity in their organization. So what are the main structures that are present in prokaryotic cell? First is, you can see the cell envelope, that means outer covering. The cell envelope is the outermost covering of protoplasm of the bacterial cell. It is known to protect the cell from mechanical shocks and different injuries. Now, this outer covering has three layers. First, you can see in this picture the outermost covering, that is blue boundaries. This is called mucilage sheath. It is the most outermost layer. It is made up of macromolecules that give sticky character to the cell. Sticky character that this bacterial cell can attach to any substrate. Hmm. So it is sticky in nature. This blue color, it is called mucilage sheath. Now, when it is thick, when this mucilage sheath become thick, then it is called capsule. So you can see the labeling also. The outermost covering, when it is thick, then it is called capsule. What is the function of capsule? It helps in resisting phagocytosis. Now, what is phagocytosis? Do you know? Acha. Can you tell me, anyone, please, what is phagocytosis?
And the engulfing of solid food by changing the structure of the cell membrane.
Yes, that means the cell membrane, the the bigger molecules that is not diffused by the cell membrane, then cell membrane creates an invagination, that means a pouch like structure. Hmm. So through this pouch like structure, the bigger molecules that is engulfed by the cell membrane. This process is called phagocytosis. So bacterial cell It is the bacterial cell, it is very small, and there is a possibility to phagocytosis by other organisms. So bacterial cell, the outer covering is made up of tough covering which is called capsule, to resist phagocytosis by the other organisms. Now you can see the next layer, which is orange in color in the picture. Next layer is called cell wall. So cell wall is present in bacteria like plant cell. See, it is present just below the capsule. It is made up of peptidoglycan. That means one kind of carbohydrate. So this layer is rigid and solid covering that gives shape, a particular shape and strong structural support to the cell. You can see the structure, that means cell wall is very rigid and it is very thick. So its function is to help in preventing cell from bursting or collapsing. Hmm. So it allows the materials to pass in and out of the cell. Next, it provides mechanical support to the cell against gravity, and it also prevents the attack of different pathogens like viruses, other bacteria, fungus, protozoa. Hmm. So this cell wall. Next layer is cell membrane. The brown color structure in the picture you can see. So it is innermost layer of the cell envelope. It is like other eukaryotic cell. This cell membrane is semi-permeable in nature and it is responsible for the interaction of the cell with the outside environment. What's function? The function of the cell membrane, plasma membrane is it helps in the regulation of the exchange of specific material between the cytoplasm and the extracellular medium. It selectively permits particular molecules to pass and prevents others. That means it is semi-permeable. So it prevents loss of components from the cell through leakage. So these are the main functions of cell membrane in bacteria. Now, in the picture, you can see the thread-like structure that is uh, red in color, thread-like structure. It is the genetic material. And in case of bacteria, that genetic material is called nucleoid. So this nucleoid is not bounded by any membrane like eukaryotes. In eukaryotes, the genetic material remain present inside the nucleus. But in case of bacteria and prokaryotes, the nucleoid, that means genetic material, is naked. In many bacteria contain a small circular DNA which is known as plasmid. You can see in the small circular DNA, that means in purple color, small circular DNA is present. So this is called plasmid. It is also a chromosomal or genomic DNA. So what is the feature of these plasmids? These plasmids contain unique characters to the bacteria like antibiotic resistance. It is also acts as sex factor. That means sometimes it acts as male, sometimes it acts as female. So sex factor. So this plasmid is very important in case of bacteria. There, this plasmid acts as antibiotic resistance. So these are the genetic material in case of bacteria. Next, other structures like prokaryotic cells lack the complex membrane-bound organelles such as chloroplast, mitochondria. These are not present in prokaryotes. But some special membranous structures are found in them such as mesosomes and chromatophores. Now, what is mesosome? You can see their cell membrane. So these mesosomes are formed by the extension of the plasma membrane into the cell in the form of vesicles or tubules or lamellae. Hmm. So this cell membrane that formed by the extension of the plasma membrane, this mesosome is formed. These mesosomes are equal to the mitochondria as eukaryotes. So their main function is uh, participate in aerobic cellular respiration in prokaryotes. It helps in respiration, then cellular secretion. It also helps in increasing the enzymatic content and surface area of the plasma membrane. So it increases the surface area of the plasma membrane and helps in respiration. It also helps in the formation of cell wall. It also helps in the replication of DNA and distribution of genetic material to the daughter cell during fission. That means cell division, during cell division, the bacterial cell that produce two daughter cell. So this mesosome helps in the distribution of genetic material. That means this DNA. Okay. Next structure is chromatophores. They are another membranous structures that are present in some prokaryotes like cyanobacteria only. Hmm. So their function is which possess photosynthetic pigment. In case of cyanobacteria, chromatophores are present as photosynthetic pigments. So they are so they are able to perform photosynthesis. These pigments are light reflecting. Now in this picture you can see bacteria can be motile or non-motile. So bacteria possess one or more thread-like appendages that extending from their cell wall. So this structure is called flagella. Hmm. So you can see the red color structure that is arises from their cell membrane. It is called flagella. Now when you uh, when you differentiate this flagella, it can be divided into three parts. You can see it is a flagella. This flagella is divided into three parts. First is filament, the longest portion extending from the cell surface to the outside. It is made up of protein called flagellin. This is filament. Then next is hook, the green color. Uh, you can see hook is a curved and tubular structure made up of protein subunits. Next, the base. It is called basal body. It is the most complex part of flagellum. This base remain buried in the cell membrane. So, what is the function of this flagella? They help in locomotion. That means movement from one place to another place. Next is pili or fimbriae. So these pili, you can see some thread-like structures that are present all around their outer covering. So these are called pili. These are also surface structures but do not play any role in locomotion of bacteria. The pili are the elongated tubular structure made up of special protein called pilin. What is the function of this pili? The pili helps in forming conjugation tube during transfer of genetic material between donor and recipient cell. That means during sexual reproduction in bacteria, conjugation tube is formed between two bacterial cells, and through this conjugation tube the genetic material transfer from one bacteria to another bacteria. So this is their sexual reproduction. So this pili helps in the formation of this conjugation tube during sexual reproduction. Now another structures that are present in bacteria is called ribosomes. Like eukaryotes, ribosomes are also found in prokaryotes and act as a site of protein synthesis. We all know the ribosome is the main site where protein synthesis takes place. So in the bacterial, that means the prokaryotic ribosome is different from eukaryotic ribosome. The prokaryotic ribosome are of 70S type. That means they possess two subunits. The smaller subunit is 30S and the large subunit is 50S. Here 30S and 50S. S means S is a Sedberg unit. That means by this unit we measure the movement capacity of this ribosome in the centrifuge machine, centrifuge tube. So this is Sedberg subunit. So that is the all the there is the structure of bacteria, that means prokaryotes. So is it clear that structure of prokaryotes?
Yes, ma'am.
Okay, some important questions from this topic, from bacteria, is that which is known to be the longest cell in human body? Do you know that? আচ্ছা এটা বলো,
Nerve cell.
nerve cell, fine. So where is uh, capsule is found?
I mean bacterial cell.
Yes, the outermost tough covering of bacteria is called capsule. আচ্ছা আরেকটা বলো, what is the name given to the infolding of plasma membrane in prokaryotic cell? Infolding of plasma membrane?
Mesosomes.
Mesosome. What is the function of mesosome? The main
And
function?
production of DNA.
Not production of DNA. It acts as mitochondria in prokaryotic cell. That means respiration. One function is respiration and another function is distribution of genetic material in daughter cells after cell division. So that is the main function. আচ্ছা আরেকটা বলো, name the only organelle that is found in prokaryotic cell. Which cell organelle is present in prokaryotic cell? Is it mitochondria, chloroplast? Okay, can you hear me? Okay, tell me one thing. So to tell me the name of one organelle that is found in prokaryotic cell.
And mesosomes.
Sorry, আবার বলো। Plasmid is DNA. It is a DNA structure. Plasmid is not cell organelle. That is not.
And mesosomes.
No, it is not mesosome. It is ribosome. Okay. So like eukaryotes, ribosomes are also found in prokaryotes. It is a site of protein synthesis. বুঝেছো? তাহলে this cell organelle is present in ribosome. হ্যাঁ, কারণ দেখো in case of prokaryotes the DNA material which is naked, it is not proper cellular organelles. এটাকে cellular organelles বলা যায় না। আচ্ছা তাহলে এইটুকু তো বুঝলে। আচ্ছা রাইবোজোমটা কি তোমরা বুঝতে পারোনি, আর একটু বলার দরকার আছে কি?
না ম্যাম, ঠিক আছে।
আচ্ছা আচ্ছা, আরেকটা কোশ্চেন, তোমরা এটা কি জানো, হোয়াট ইজ গ্রাম পজিটিভ ব্যাকটেরিয়া, হোয়াট ইজ গ্রাম নেগেটিভ ব্যাকটেরিয়া?
ইয়েস ম্যাম।
হোয়াট ইজ গ্রাম পজিটিভ ব্যাকটেরিয়া? হুম, ওকে, সো এক্সাম্পল।
ম্যাম, ভিব্রিও কোলেরি।
ভিব্রিও কোলেরি না, এটা হচ্ছে বেসিলাস, তারপর ক্লস্ট্রিডিয়াম। হ্যাঁ, আর গ্রাম নেগেটিভ ব্যাকটেরিয়া হচ্ছে হুম হুম এসেরিকিয়া। গ্রাম নেগেটিভ ব্যাকটেরিয়া, that means the bacteria that do not take up gram stain. These are called gram negative bacteria. ঠিক আছে। আচ্ছা, নেক্সট পোর্শন হচ্ছে কি, ইউক্যারিওটিক সেল। তো আমরা প্রোক্যারিওটিক সেল পড়েছি, এবার ইউক্যারিওটিক সেল। সো দেখো, a cell which has a well organized nucleus with a nuclear envelope and several membrane bound organelles is called eukaryotic cell. That means the eukaryotic cell have eukaryotic cells have cell wall, proper cell wall, and then vacuole, cytoplasm, ribosome, plastids, Golgi apparatus, mitochondria, etc. Now, eukaryotic cells are found in all protists, plants, fungus, all animals. Hmm. Eukaryotic cell is larger than the prokaryotic cell. এবার all the eukaryotic cells are two types. One is plant cell, another is animal cell. So there are some basic differences between plant cell and animal cell. So here is the structure of plant cell and an animal cell. You can see cell wall is present in plant cell, but it is absent in animal cell. Then vacuole is present, large central vacuole is present in plant cell. But in case of animal cell, it is the vacuole is one or more, but the size of the vacuole is smaller than the plant cell. Then another difference is plastids. In animal cell, plastids are absent. In plant cell, plastids are present. Then other structures are almost similar. That means mitochondria, Golgi apparatus, endoplasmic reticulum, ribosomes, all these are present in both eukaryotic cell, plant cell and animal cell. Now we are going to discuss all the cell organelles in detail. Now, so some cell organelles are common in both plant cell and animal cell. For example, cell membrane. So every living cell is covered by a thin, elastic, transparent, semi-permeable membrane, which is called cell membrane. This cell membrane is also called plasma membrane or plasma lemma. The plasma membrane separates the internal environment of the cell from external environment. The membrane helps in regulating the entrance or exit of molecules into and out of the cell. It regulates the movement of substances. Now, structure of cell membrane. See, the cell membrane is composed of lipid bilayer with protein molecules embedded in it. Sometimes carbohydrates are also present in cell membrane. Now, it is a model that is represented by scientists Singer and Nicholson in 1972 to study the lipid and protein uh, structure of the cell membrane. So this mosaic-like arrangement of lipid and protein is called fluid mosaic model of Singer and Nicholson. So according to this model, the lipid molecules are here two types of lipid molecules are present in the cell membrane. First is polar hydrophilic end. Polar end. So this polar end, that means this region is the form of head which faces towards the outer side of the cell membrane to interact with the aqueous environments on both sides. That means polar head. First হচ্ছে কি lipid structure. Lipid structure, when you describe the lipid structure, so lipid structure has two end. One is polar hydrophilic end and another is non-polar hydrophobic end. So you can assume hydrophilic means water loving ends. So this region, that means this polar hydrophilic head which faces towards the outer sides and of the cell membrane and inside the cell membrane. These interact with the aqueous environments on both sides. And another end is called non-polar hydrophobic end. So this region is called water repelling tail. Both ends of which faces each other that occur towards the center of the cell membrane. You can see the tail-like structure. This is called hydrophobic tail of lipid bilayer. Both the hydrophobic end faces each other and that occur towards the center of the cell membrane. Now, protein structure of the cell membrane. The you can see the some protein molecules are present. Some protein molecules are floating in this lipid bilayer structure. Now the ratio of protein and lipids, it varies considerably in different types of cell. But in case of RBC, that means human red blood cell, the protein consists of 52% and lipid consists of 40%. Now this membrane protein can be classified as two groups. One is integral proteins, another is peripheral proteins. So integral proteins means they have stronger association and with membrane, and these proteins are buried partially or totally in the phospholipid bilayer. So some proteins are there, they are partially or totally buried in the phospholipid bilayer. These are called integral proteins. And another types of proteins are there which is called peripheral proteins. So peripheral proteins have weaker association and they are bound to lipids of membrane by electrostatic interaction. Now some carbohydrate structures are also present, that means 1 to 5% of their chemical composition. Carbohydrates also present in plasma membrane. These are associated with the phospholipids. That means with the lipids, these or with the peripheral proteins to form glycolipids and glycoproteins respectively. That means the carbohydrate chain, you can see the blue color chain is present, these are carbohydrate chain. So when this carbohydrate is attached to the protein, they form glycoproteins. When this carbohydrate chain attached to the lipid, then it is called glycolipids. Now this membrane, plasma membrane, has the ability, that means the proteins, the protein can move from one place to another place in this lipid bilayer part. And it is the nature of the membrane is fluid, because it functions like intracellular junctions, then cell growth, secretion, endocytosis, and cell division. This cell membrane is fluid in nature because it has to perform different functions such as cell growth, then secretion, endocytosis, cell division, etc. Now, this function of the cell membrane. Cell membrane performs following function. It is selectively permeable or semi-permeable membrane and allows only selected substances to pass in. It protects the cell from injury. Then the membrane have carrier proteins for active transport. এবার active transportটা আমি তোমাদেরকে বলবো। Next functionটা হচ্ছে কি, cell membrane contains different enzymes which perform certain reaction on their surface. Enzymes like ATPase, phosphatase, these are present on the cell membrane. So these are the main functions of cell membrane. Now, the main function of cell membrane is transportation of substances. Passage of substances across the membrane occurs mainly by two methods. One is active transport and another is passive transport. Now, এখানে important question আসে, differentiate between active transport and passive transport. Now, you need to know what is active transport. So, active transport is the movement of molecules across the membrane against their concentration gradient. So, what is concentration gradient? Concentration gradient means when the substances move from their lower to the higher concentration. So, from normally we know when any molecules that move from higher concentration to their lower concentration. But in case of active transport, the substances or molecules move across the membrane from lower to the higher concentration. That means against their concentration gradient. It is an energy dependent process, as it is the membrane against their concentration gradient, so it needs energy, in which ATP is utilized. ATP provides the energy for the transport, that means active transport. Some ions such as Na plus, K plus pump also takes part in active transport. Now, some molecules, for example, polar molecules require carrier protein of the membrane to facilitate their transport across the membrane, because they cannot pass through the non-polar lipid bilayer. The substances, for example, some polar substances, they cannot pass through the non-polar lipid bilayer. So these substances transport by carrier protein. You can see the protein molecules that are present in the lipid bilayer. So this protein, integral protein, they act as carrier protein for polar molecules. Now, so it is active transport. Next is passive transport. Passive transport is the mode of movement of molecules or substances across the membrane without any requirement of energy. So, by three ways, passive transport can occur by the cell membrane. First is osmosis. So, what is osmosis? Osmosis is the process by which water molecules pass through a membrane from a region of higher concentration to a lower concentration. So, water molecules, when pass through the membrane, this is called osmosis. Another passive transport process is simple diffusion process. Diffusion process means neutral molecules move across the membrane along the concentration gradient from higher to the lower concentration. For example, gases and small molecules by diffusion process. Next is facilitated diffusion. In this process, the molecules are transported along the concentration gradient by the help of ion channels and permeases. Energy is not required in this process. By the ion channel, some ion channels, that means such as Na plus, K plus, pump. So these are act as ion channels. So some molecules that use these ion channels to move across the membrane. So this type of movement is called facilitated diffusion. Energy is also not required in this process. So this is a kind of passive transport. Okay, so here you need to learn the difference between active and passive transport, and what is osmosis, what is diffusion, facilitated diffusion. So these are the important question from this topic. ওকে, আজকে তাহলে শুধু সেল মেমব্রেনটাই ডিসকাস করি, বাকি সেল অর্গানেলিস এটা আমরা গ্রাজুয়ালি নেক্সট ক্লাসে আমরা ডিসকাস করব। আচ্ছা, যে সেল মেমব্রেনের যে স্ট্রাকচারটা বলা হয়েছে, এটাতে কোনো কনফিউশন আছে তোমাদের? ফ্লুইড মোজাইক মডেল অফ সিঙ্গার এন্ড নিকলসন।
নো ম্যাম।
আচ্ছা, ফাইন। আমি এই আরেকটু তোমাদেরকে বাকি সেল অর্গানেলিস গুলো আগে বলে দিই, মানে বুঝিয়ে দিই, তারপর এখান থেকে কোশ্চেন দিয়ে দেব। আচ্ছা, নেক্সট লাস্ট ক্লাসে আমি বলেছিলাম তোমাদেরকে কিছু কোশ্চেন দিয়ে দেব, অ্যানিমাল কিংডমের কিছু পোর্শনের মানে কোশ্চেন দেওয়া হয়নি। আজকে আমি এটা তোমাদের কোশ্চেনটা দিয়ে দিচ্ছি। হ্যাঁ, এই কোশ্চেনগুলো তোমরা একটা কপিতে করে নেবে। হ্যাঁ, তোমাদের কারণ নোট কপির জন্য তোমাদের কিছু আলাদা মার্ক আছে। ঠিক আছে, তাহলে প্রত্যেকদিন আমি ক্লাসে যে কিছু কোশ্চেন দিয়ে দিচ্ছি, এগুলো কিন্তু তোমরা কপিতে তুলবে, যখন স্কুল খুলবে আমাকে দেখাবে। আমি সাইন করে দেব। ঠিক আছে, তাহলে আজকে এখানেই শেষ করছি। হ্যাঁ, আজকে যে ক্লাসটা করানো হলো, তোমাদের কিছু ডাউট আছে, কিছু ডিসকাস করতে হবে?
নো ম্যাম।
ঠিক আছে, ফাইন, তাহলে আমি কোশ্চেনগুলো দিয়ে দিচ্ছি, গ্রুপে চেক করে নিও তোমরা। ওকে।